The first week following insemination is a period of major embryo mortality in passive gas. There is high implication for research direction นะคะก็จะเป็นในเรื่องของการศึกษาด้านการพัฒนาตัวอ่อนแล้วก็มาดูว่าทำไมหลังจากการที่เราผสมเทียมไปแล้วเนี่ยทำไมถึงมีภาวะการล้มเหลวของในคันทองนะคะโดยใช้ในวัวนะคะเป็นต้นแบบให้กับสัตว์ป่าชนิดอื่นๆค่ะดร. เดบรา เบิร์กเนี่ยท่านทำงานอยู่ที่สถาบันแอนทริเซิร์นิวซีแลนด์นะคะท่านก็เป็นผู้เชี่ยวชาญในเรื่องของการผลิตตัวอ่อนพัฒนาองค์ความรู้ด้านวิทยาศาสตร์มาตั้งแต่สมัยท่านเรียน PhD นะคะโดยเฉพาะในสัตว์ป่าตัวนะคะ The floor is yours. Please welcome Dr. Debra.
Good afternoon. I hope everybody's not too tired after lunch. I know you're all sitting here thinking, now what on earth do dairy cows have to do with deer? First of all, I don't do deer work much anymore. It's a very small part of my research program. But I promise you, even though I changed the title on and the topic, I will bring deer into the story at the end. But you actually need to have this first part of this story before you can understand the rest of it. So what we're going to really talk about today is for some of you PhD students, you design an experiment, you look it through the literature, and you're going to expect a result. And you know what you're going to get before you get started. Well, that's how we started this research project. We figured we knew what the answer was before we started, and we had some surprising results. And I'm going to tell the story on how we had to save the program. So the outline I'm going to talk about is the conception loss trial, the background, trial design, embryo survival, conception loss pattern, and some things associated with that, and then our change of research direction. And I have two PhD students who looked at IBM medium based on cow follicular fluid and the search for uterine biomarkers. And these are the things that we're going to bring in for the Ellis Deer project, or those two projects at the end. In New Zealand, dairy cows have a very, very short breeding season, only 6 to 9 weeks. That's very, very similar to deer. Our dairy cows are seasonal. Compact breeding season, so we have a compact calving pattern, so that the peak grass matches peak lactation. That's almost the same thing as what you have in deer. They have the same thing, only instead of giving the milk, they have to feed the calf. So it's actually very, very similar. We have a 12% gap between how we measure reproductive success in dairy cows, which is 6 weeks, calving rate. The industry target is 78%, and ours hovers anywhere between 64 and 68%, and it has not changed in the last 10 years. The worst part is that we have an empty rate, because the animals who don't get pregnant within that 6 to 9 weeks, then they're empty, they're not bred, the farmer either has to carry them over or he has to cull them from the herd, and that's very expensive as well. And we have a gap of 13%. So our research problem was to actually find out what's happening. Conception loss in dairy cattle. This is a pie chart, and this data is probably older than the majority of you in this room. It's 40 years old. This data was collected in the late 70s and the early 80s, and it has been perpetuated all this time. And people think that in seasonal, moderately milking dairy cows, fertilization failure is not a problem. It's right around 90%. The greatest loss occurs in the first 3 weeks. After insemination, which is 25%, we have very few loss after that. We have something called phantom pregnancy, and that is when the embryo elongates and it signals a pregnancy, but there's actually no embryo, and it's lost between day 28 and 42. Then we have fetal loss, which is losses after day 42. The period of embryonic development is from insemination to 42 days in cattle. And the calves born is 55% rate. That hasn't changed much over the years. The thing that drove research for almost 40 years in this area is they found that the greatest loss was between day 8 and day 16, which is the maternal recognition of pregnancy. And that's why if you read dairy cattle embryos, papers in the last 20 years especially, that's all you're going to read about. So we wanted, in order to solve the research problem, we had to know when the actual embryo was lost. And we wanted to quantify the losses up to day 70, and we also wanted to associate anything on farm that we could associate embryo loss with, so we know where to start. We had 4 collection groups, and they were designed to encompass the major developmental programs. So we flushed embryos out at day 7, we flushed embryos out at day 15. We had another group where we used ultrasonography, and we evaluated pregnancies at day 28 and day 35, and then we also had some controls, which is what we do on the farm, and we fetal aged the cattle at day 70 with ultrasonographies. This is a randomized complete block design. We enrolled 2,000 cows over 4 farms, and we did this over 2 years. It's a huge trial. We flushed over 1,000 embryos. So you can imagine the scale of that type of a project. This is our embryo survival. Now, embryo survival does not include conception rate, fertilization failures. It's just the embryos at about day 1.5, we started 100%. And if you can look, our day 7s, you can see that we lost about 24% from insemination to day 7, and you can see that statistically there's no difference between the embryo loss at day 7 until you get out to day 70. That's incredible. What happened to that major embryo loss between day 8 and day 15? We didn't have it. And you can see that the major loss occurs in that first 7 days. Now that was the surprise. It's also a really difficult period to study. I'm going to skip the next slide because it's a little bit confusing, and I'll talk to people afterwards if they want to talk about it. One of the things that we did find that was associated with embryo survival is the choice of the bull. It actually affects conception rate. We had 40 sires throughout this whole thing, and we were able to we used sire as a random effect, and we found that sire made a difference, but it only explained about 8% of the variation in conception rate. We used logistic regression to look at these things, and what I want to make perfectly clear is on the vertical axis, that's not pregnancy rate, that's probability of conception. Those are 2 very different things. So you can see that first circle there, that was our highest bull, and he had an 82% probability of giving a very good conception. Now that's compared to something down here with this bull, it's less than 50%. So you know when you have a calving rate of 55% and you use bulls, the probability of conception is less than 50, it's not very good. The thing that I really want to stress here too as well is this is the actual conception rate for the bulls. It's not its genetic merit. A lot of these bulls here, their transmitting ability to their daughters for fertility is actually quite good, but their own fertility themselves is not. We also found that increased conception rate was it was associated with days postpartum. So increasing days postpartum, we had increasing conception rate. And again, logistic regression, we can take the probabilities and we can say for certain that we have an extra 13% probability of a successful conception with every extra week postpartum. If you take that on an estrous cycle basis, cow 21 days, 3 weeks, you increase your probability of conception 43% if that cow has an extra estrous cycle. We also look at estrous cycles as well, and that is equivalent to an extra day 8 postpartum. And the probability of conception increased by 9% when the day 7 progesterone was raised from 3 nanograms per mil to 7 nanograms per mil. We've also done some mathematical modeling on early progesterone, and we found that in our system with our assay, code count assay, which is no longer available, the optimum day 7 progesterone is 7.8 nanograms per mil. Now the interesting part about this is days postpartum, estrous cycles, and increased progesterone. That was all known before, so that's not new information. The new information is that we actually can see this effect as early as day 7. Now that's very, very different. This is usually associated with pregnancy rate anywhere between day 35 and day 70, but we can actually associate that with a day 7 conception. So this is the conclusion to the trial, and like I said, it was very, very surprising for us that the major embryo losses occur within the first 7 days. Recognition of pregnancy in the New Zealand dairy cow is not a problem. Now that's one of the things that I've been studying for the last 10 years, so that was actually quite a surprise for me. And fertilization rates, I didn't report them, it was 83%, and that's similar to the North American Holsteins when you inseminate under thermal neutral conditions. When you inseminate under heat stress, then it can go as low as 40%. Choice of sire really affects conception rate, and that's something that New Zealand has never measured before. The States has actually started to measure it, and they put it in their fertility index, but New Zealand has not. And again, we talked about postpartum progesterone and estrous cycles. But the thing I want to stress here, especially for the PhD students, is that the result we got was not what we expected. And we had a lot of research planned, future research based on we were going to lose embryos from day 8 to day 16, and we did not. Day 7, the losses between insemination and day 7 are incredibly difficult to study in vivo. In vitro, it's very different, but in vivo, it's difficult. Is it the ovary? Is it the follicle? Is it the oocyte? Is it the oviduct? The uterus could be involved. They all could be involved, and they could all have a little bit of a conception loss. Some cows may have problems with ovary development. Some may have problems with just the uterus. And so sorting it out is actually quite difficult. But that was what the industry wants, and that's what we're doing. So now I'm going to just present a couple research projects that I found incredibly interesting, and I think they have real application for the Eld steer. So now we're going to switch. What we did is we decided that we needed to study in vivo oocytes, but we wanted to study them in vitro to see if we could figure out what was going wrong. So what we did is we created a physiological relevant IBM medium based on dairy cow follicular fluid information. And this is a PhD student's work. Her name is Latasha Murray. And so everything that I'm going to present today is Latasha's work. And this has also been a co-project with Dr. Janet Pittman down at Victoria University. What we did is we analyzed follicular fluid from antral follicles, from dominant follicles, and from pre-ovulatory follicles. And we devised 2 maturation media. One you can see here in green, that was the good media. So what we did is we took all the concentrations, and the concentrations that were in the upper 10% quartile, that is what we used for the good media. The bad media on the other side was the lowest concentrations, and we devised the media of that. So the concentration is the only thing that's different between the good and the bad media. We also have amino acids. Amino acids are quite interesting because amino acids in New Zealand dairy cows are only about 70% of what they reported in the Irish, and about 50% of what they reported in North America. Everybody always said that when we measured amino acids, our assay wasn't very good because we had lower results. What this graph shows is in green, it shows the concentrations of amino acids that we used for the good media, and in the red is the concentrations of amino acids that we used in the bad media. The black dots are the concentrations of amino acids that you'll find in our standard M199 media. And you can see that most of them are higher than what we normally need, and cysteine is actually much, much lower. And that's why people actually add cysteamine to their maturation media. But you can see that the amino acid concentration is very different. And poor Natasha made amino acid stocks, and she composed this media. And I want to tell you guys, it was 120 components that she used. She had patience. Now, if we're going to follow oocytes through, what we had to do is we had to devise a single in vitro embryo production system. We always have them in groups because oocytes and embryos have paracrine and autocrine effects, and it helps the other oocytes. We had to do this in singles because we wanted to follow the same oocyte through. We had 3 media and sometimes 4. We had our good media, we had our bad media, we had our control media, or the commercial media, which is just M199 fetal calf serum, and we had single and group cultures. We used a biphasic media. Our early medium had a meiotic inhibitor in it, and that was to try and get cytoplasmic maturation going. We used that for 8 hours. Then for the additional 22 hours, we had our late medium, which was based on preovulatory follicles, and we added LH to those. After IVM, you can see here we actually did a cumulus cell biopsy before IVF, and we cut those with a knife. And this was biopsy for gene expression, and this was put in for IVF. This graph just shows the effect of IVM medium on blastocyst rate and quality. As you can see, there isn't any difference between the commercial media M199 or the good media, which is in green. But it had a huge effect on blastocyst rate with the bad media. I also want to call your attention to that this is actually single oocyte culture, so having a 35% blastocyst rate was pretty good. We were pretty impressed with that. But we really did have a media effect on blastocyst rate. Blastocyst quality, it tended P equals 0.06. Those are always really, really frustrating for scientists. It's almost significant. And you can see that the good media actually had an effect on blastocyst quality. Much higher quality than either commercial media or the bad media. Now this next graph, we counted cells. You know that a blastocyst has got the inner cell mass and it has trifectoderm. And we differentially stained them and we counted them. And what you can see is that there was actually no difference in the cell numbers in the trifectoderm or the ICM with the 3 different IVM medias. But what we did see is we saw an effect on quality. There was less pycnotic cells in the blastocyst when we used the IVM good media, and that's also true in the TE as well. So the good media, actually the blastocyst quality, we saw it, we trended with it, and you can see here that the pycnotic cells are significantly lower. I'm just, like I said, I'm cherry picking a bit of these results, and what I want to call your attention to is that blastocysts in the graph are on the left-hand side and all the rest of the embryos are on the right. This is the results of gene expression on the cumulus cells. And we have 4 genes here that I'm talking about today. The first 2, there was an effect of media, and there was an interaction between media and outcome. And the genes that we used was GA1, which is a gap junction protein, and that's associated with COC quality. And NRP2, which is an antioxidant pathway, and that's also with embryo quality. What I want to call your attention to is the fact that in the blastocyst, the gene expression, you can see that the group cultures that we normally use are the hatched bars. And you can see that the good media and the group culture has similar gene expression in these upper two boxes, or graphs, sorry. And that to me was really, really nice. It shows that even though we've got single culture, the good media and the bad even as well is the embryos aren't struggling as much, we don't think, as it is in the single culture media. Then we also found two genes where we could actually see a difference in outcome. And that was PDE8, and that was very satisfying because that's involved in meiotic resumption, and that's linked to genetic fertility in dairy cows, and BMP receptor 2, and that regulates granulosa proliferation and inhibits apoptosis. And both of those had higher gene expression in blastocyst rate. So they're potential biomarkers. They have to be validated. They haven't been validated yet. So I wouldn't go right out and test them yet. So in summary, our IBM bad medium based on the lower 10% concentrations negatively affected blastocyst development, and conversely, our IBM good medium increased blastocyst quality. We found a couple cumulocell gene biomarkers, and they were associated with embryo developmental outcome. So this, to me, is something that we could do in the old steer, because we could actually look at the follicular fluid environment and maybe make an IBM media specifically for old steer. I did that with the oviduct in red deer, and that's the media we're using now. But this next bit of information is, I'll explain it because technology has really moved forward, and I think it's quite feasible. This is my other PhD student. This is Nick Arena, and we're actually doing multi-omics, and we're investigating biomarkers for uterine suitability in early pregnancy. And this is all preliminary data at this point, and I'm going to go through this quite quickly. The reason we're interested in uterine proteins and metabolites is because we had that increasing conception rate improves over time. And so that uterus is actually changing over time is what our hypothesis is. And the thing is, what I want to explain is that our random variation in our models, the cow could explain 87% of the random variation in the model. So we know the cow is our biggest variable. So how do we cut down on the cow itself? This is in the wrong order, but that's all right. This is just a schematic of how we're doing uterine flushing and how we take the samples from metabolomics and proteomics. You don't really have to understand what's going on. It's just as that we put 30 mils of flushing media into the uterus and we put it back. We've got a tracer. It's a heavy tracer, and that's so we can normalize proteins. We use proline. So what we have to do is after we collect it, we have to spin out all of the extraneous cells, and we use the filtrate and the retentate, and we do extraction from metabolomics and protein extraction to proteomics. And so we're using the same samples. This is what I wanted to talk about. Because we have cow variation, what we're looking at is we're looking at cow uterine proteins and metabolomics on their first expressed estrus, and then the same cow again on their third expressed estrus. And that's because we're going to get a very different pattern. We have some cows that give us nothing. We have other cows that is what we expect. Embryo development is not good on the first estrus, and it improves by the third estrus. We also have cows that is very good on the first estrus and not so good on the third estrus. And these are gold standard, they're good all the time. And that's how we're going to do the comparisons. We're going to compare this to this, and then each individual. I just want to show what the embryo survival was, because we use the embryo as the biosensor to tell us what the uterine environment is. Is the uterine environment good, or is it very, very poor? And the embryo is the one who can tell us that. And you can see here, the first estrus is in blue, the third estrus is in green. You can see that fertilization rates improve as we go from the first estrus to the third. 83% is what we had for our trial, and we're up to 95% on the third estrus. And that's because we used the semen from the bull that gave us the 83% probability of successful conception. Now, everything on the right of that black line is embryo survival, and of course, embryo survival is 100%. You can see that by day 4, those are embryos that are rested at the 16-cell stage or less. That's in the oviduct, and you can see that that does not change over time. It hovers right around the 10% loss. Here we see a difference of about 12% between on the uterus, between the first estrus and the third estrus. So this is what we're going to be looking for, that difference of 12%, and we think that this gives us a real biological variation instead of looking at between cows variation. This is the most exciting part of the whole thing. It's rapid evaporative ionization mass spectrometry, and we call it REAMS. This was developed for cancer surgery. What it is a mass spec attached to a scalpel. And what the surgeons do is when they have a cancer tumor, they actually cut the tumor out and they use the mass spec to get a fingerprint of the cancer cells, and then that fingerprint will change when they cut into healthy cells. So what you have is you've got a mass spec fingerprint between the two, and you can differentiate them. The other thing is, this is real time, people. This is cool. You don't have to sit and prepare samples for a week and then run them on a mass spec and it takes you days. This is in real time. And research has one of these. And one of the most interesting things about this is we've been working with the engineers, and we've got it to work in uterine fluid. Uterine fluid that we take right out of the cow and we spin the cells out. This is a PCA plot, and it is based on embryo quality, excellent, good, poor, no embryos. And I know that the graph isn't very good, but this was Nick's first attempt at PCA, and it's only 20 samples. But he analyzed those in less than an hour. And he had 1,000 compounds that he could actually identify. He can't identify them, it's just a fingerprint. So there's no identification of the actual samples of the peaks themselves. It's just a fingerprint. What I want to show you is you can see that the excellent quality is on the far left, and those have grouped quite nicely. And we've also got a grouping with the no embryos. The no embryos are interesting because we've got a technical, we're flushing the embryo out, it's 150 microns, and it's an great big uterus. And we could have missed it. So we're hoping that we can get a fingerprint to tell us whether there's an embryo or not. This is really exciting for me. So it has the potential to find uterine biomarkers in the unprocessed uterine fluid. And the other thing I didn't mention was the proteomics, but we've actually taken that and we've actually increased it from 80 proteins to about 200. And the key there is get your right, select your choice of databases. So the implications for red deer, or for Eld's deer, is I think that we would benefit immensely if we could actually use physiologically relevant IBM media. We could collect the follicular fluid using electroscopic OPU, and we could, and the amount of fluid is dependent upon the assays. But because the technology is changing so rapidly at the moment in the metabolomics area, we probably could get enough fluid to actually get a very, very comprehensive sample. Human follicular fluid has been analyzed and they're correlating it to pregnancy rates now. So those I've listed amino acids, glucose, estradiol, progesterone, LH, FH, FSH. I think those would be the absolute minimum we would try. But with this new technology, I think that maybe we could really go for the gold standard and actually have a really good IBM media based in deer. And the other thing I just would like to say is because we know conception rate increases with time, please, when you see those recips, make sure they're got a seal, please. And as for everything, I'd like to thank our funder, New Zealand Ministry of Business and Innovation, pillars of the sustainable dairy industry, and you can see that there's an awful lot of people involved in this. So thank you very much.
เป็นงานวิจัยที่ลงลึกรายละเอียดนะคะอันนี้ก็จะทำให้เห็นว่างานทางการเอามาประยุกต์ใช้นะคะที่จะทำให้ได้จริงเนี่ยมันต้องผ่านกระบวนการการศึกษาวิจัยมาค่อนข้างมากนะคะจริงๆอย่างที่บอกว่าดร. เดบ้าเนี่ยชำนาญในเรื่องของการศึกษาเรื่องการพัฒนาตัวอ่อนกวางนะคะแล้วก็เรียนเชิญท่านมาบรรยายในเรื่องของตัวกวางนี้นะคะแต่ว่าท่านบอกว่าปัจจุบันเนี่ยที่นิวซีแลนด์เราพบปัญหาเรื่องของการใช้ฮอร์โมนนะคะสมัยก่อนเราเข้าใจว่านิวซีแลนด์เนี่ยส่งออกเนื้อกวางนะคะเป็นอันดับต้นๆนะคะแต่ว่าปัจจุบันเนี่ยเขาพยายามที่จะไม่ใช้ฮอร์โมนในการใช้ผลิตตัวอ่อนนะคะก็เลยทำให้ท่านเลยต้องเปลี่ยนเรื่องของหัวข้อการวิจัยมาศึกษาในเรื่องของธุรกิจปากโคนมมากขึ้นนะคะแล้วก็ตัวอย่างที่ท่านพยายามที่จะยกขึ้นมาให้เห็นเนี่ยก็จะแสดงให้เห็นว่าศึกษาค่อนข้างมากแล้วก็อาจจะเป็นไปได้ว่าจะพัฒนามาใช้ในสัตว์ป่ากับกลุ่มกวางในเรื่องของการอนุรักษ์นะคะก็ขออนุญาตสรุปคร่าวๆนะคะในเรื่องของที่ท่านได้กล่าวมาตอนแรกนะคะก็จะเป็นว่าปัจจุบันเนี่ยทำไมธุรกิจการผลิตโคนมเนี่ยมันก็ทุกคนมีความต้องการมากขึ้นนะคะแต่ว่าความสามารถในการขยายพันธุ์ของโคนมนะคะในประเทศนิวซีแลนด์เนี่ยมันลดน้อยลงนะคะท่านก็เลยศึกษาในช่วงแรกนะคะว่าภายหลังจากการผสมเทียมไปแล้วเนี่ยมันเกิดปัญหาอะไรนะคะท่านก็พบว่าในช่วง70 วันแรกนะคะภายหลังการผสมเทียมเนี่ย 67% นะคะมีการสูญเสียของตัวอ่อนนะคะโดยเฉพาะช่วง 1-7 วันแรกนะคะที่มีการหลังจากการผสมเทียมเข้าไปอันนี้ก็จะเห็นแล้วว่าขณะเรื่องของการผสมเทียมนะคะก็ยังมีปัญหาอุปสรรคค่อนข้างมากนะคะท่านก็ให้ข้อแนะนำแล้วก็เปิดประเด็นว่าในอนาคตเนี่ยจะต้องมีการศึกษาอะไรต่อไปบ้างนะคะโดยเฉพาะเรื่องของข้อนะคะที่จะมาใช้ในการผสมเทียมตัวนี้ก็จะเห็นได้ชัดเลยว่าจะเป็นปัจจัยมาที่ส่งผลต่ออัตราการตั้งท้องนะคะรวมถึงเรื่องของงานที่ยังไม่ได้มีการศึกษานะคะเช่นกระบวนการส่งผ่านข้อมูลระหว่างแม่กับลูกนะคะก็จะเป็นปัจจัยที่สำคัญนอกจากนี้นะคะท่านก็ได้พูดถึงเรื่องงานวิจัยต่างๆ Sorry it's quite long but I try to make it short เรื่องของน้ำยาที่จะนำมาใช้นะคะในการผลิตตัวอ่อนนะคะท่านก็ได้ทดสอบกระบวนการแล้วนะคะในเรื่องของอะมิโนแอซิดนะคะระดับอะมิโนแอซิดว่ามันมีความสำคัญมากนะคะยิ่งทำให้เหมือนกับธรรมชาติได้เท่าไหร่เนี่ยมันก็จะยิ่งดีน้ำยาที่มีการปรับปรุงนะคะได้ใกล้เคียงกับธรรมชาติก็จะยิ่งดีมากขึ้นนะคะนอกจากนี้ก็ท่านก็ได้พูดถึงเรื่องมาร์เกอร์ต่างๆนะคะที่เราเข้ามาใช้นะคะในการตรวจสอบคิวมูลัสเซลล์หรือว่าเซลล์ที่เป็นที่เลี้ยงไข่นะคะดูว่ามันจะส่งผลต่อความสมบูรณ์พันธุ์ของเพศเมียมากน้อยแค่ไหนนะคะแล้วก็มีการพัฒนาของตัวอ่อนดีขึ้นมากน้อยแค่ไหนนอกจากนี้ก็ลงลึกไปอีกนะคะเราก็อาจจะได้ยินคำว่ามัลติโอมิกส์นะคะก็คือโปรตีโอมิกกับเอนไซม์โพลีโอมิกส์นะคะซึ่งก็จะเป็นการศึกษาที่ลึกลงไปในเรื่องของระดับโปรตีนแล้วก็เมตาบอลิซึมนะคะท่านก็ศึกษาในเรื่องของโปรตีนนะคะในมดลูกนะคะซึ่งมันจะส่งผลต่อความสมบูรณ์พันธุ์ต่างๆนานานะคะซึ่งตรงนี้เนี่ยท่านก็ให้ข้อแนะนำว่าก็สามารถที่จะเปิดแนวให้เราพัฒนานะคะกระบวนการต่างๆเทคนิคต่างๆหรือว่าส่วนประกอบต่างๆที่สามารถนำมาใช้นะคะประยุกต์ใช้กับสัตว์ป่ากับตัวกวางได้อันนี้ก็คือข้อสรุปสั้นๆนะคะให้ท่านได้รับทราบท่านใดมีข้อสอบถามนะคะเรียนเชิญค่ะ
Actually, I have two questions. So what is your definition of embryo survival in the first graph that you showed where you collect 1.5 day post AI and then 7 days and then after that it doesn't change, but what is your definition of survival?
Okay. It's actually embryo quality, and I can go through it with you, but what we did is when we collect embryos at day 7, You know you've got embryos that, or you have unfertilized oocytes or single cells, so that would be figured would produce as fertilization failure. Anything that cleaved and arrested anywhere up to the morula stage, we consider those, they were dead, they're arrested development, because we expected to have type morula and blastocyst. So at day 7, what we had were type morulas grade 1 and 2, and blastocysts grade 1 and 2. We know that the grade 3s, and that's the conception loss graph, we know that those grade 3 TMs by the literature, we'll lose 80% of those by day 15. Only 20% of those will go on. So what we're looking at is embryo quality. So at day 15, we looked at elongation. It had to be a certain length.
Okay, at 1.5, just at the beginning, or when you have almost 100%, what did you look at? What did you get?
Why did I get 100%?
No, what is the type of embryo you get? What, 2-cell stage?
Yeah, I just took out anything that was at the single cell stage.
Okay. So the difference actually that you have between 1.5 and 7 is exactly what you see also in vitro, right? With a loss of.
Yeah.
Okay.
And it's not the same. You know, we lose a lot more in vitro.
Yes. Okay.
But yeah, but it's exactly the same. Yeah.
Okay. Okay, but to me the most interesting part is that it doesn't, after that it doesn't change, right? Okay, because, okay. And my second question was about the embryo flushing or even the orbital flushing to characterize the follicular fluid or the uterine environment. So for the uterus you say that you inject 30 milliliters of fluid and that you recover everything. So, well, you recover something that's kind of overall represents whatever is in the uterine wall and the corpus of the uterus. But if you wanted really to try to characterize more like regions by region or section by section, what would be the approach then to try to have a better idea of, because maybe the proteomic or metabolomic profile is not the same at the tip of the horn or in the uterine body, right?
Right. The uterine body in cattle as it migrates through there, that's day 15. So we can forget that. And you're right, we're just getting the overall. What we did try to do is a mini tube has got a very, very flexible catheter that they use to deep interuterine AI to get right up to the oviduct. And we actually tried to aspirate the actual uterine, for lack of a better word, I'm sorry. It's very, very viscous mucus. And so what we tried to do is we tried to get, we tried to get, we tried to get that mucus, and one of the problems with that is that to actually resuspend that mucus into something that we could actually extract, we just can't. So you almost have to take it and put it through a gel system, and that has to wait for you. So that's why we need to go through this. Sorry for the rain.
เดี๋ยวก็ได้
มีท่านไหนมีคำถามเพิ่มเติมไหมคะค่ะก็เสียงปรบมือนะคะให้กับดร.